I'm Anthony Ament. I'm in the Department of Botany at UH, and I don't study plants. I study the things that eat plants. I study fungi. And so I'm going to tell you a little bit about fungi in the mesophotic zone. I know you heard a bit about that last week when Dr. Sherwood came and talked about algae. I'm also going to do a little bit of a bait and switch because I'm going to talk about some of the other research that we've done also that led me to the mesophotic zone into the ocean. So I always go on too long, so I'll start off with my thank yous, just in case I run out of time. This is my lab group. This is at Kaimana Beach, and I have a lot of thank yous. A lot of people have funded this work. The National Science Foundation, Department of Defense, actually, the Keck Foundation most recently, and then a whole bunch of collaborators and students and friends and family that have chipped in along the way. This is Kaimana Beach. Waikiki.
And who's Keck?
Keck Foundation, they're a group, they're based out of LA, and we just got funding to do work in Waimea Valley up on North Shore. And I'll tell you a little bit about that at the end of this talk. So I'm going to start off with a slide of the very first described marine fungus. This was described in the 1840s on plants, a marine plant called Spartina, and they found this in North Africa, so in Algiers. It was two French explorers, and they found this fungus that was growing on the root of one of these plants, and they wrote it up and described it, and it was sort of a sensation because nobody had really considered the fact that there was fungi in the ocean. People had seen mushrooms growing and mold growing on land, but to find these things in the ocean was something that was sort of new. And then there was radio silence, and then nobody else really picked up on it from there. And so we have a big gap of about 100 years between this very first marine fungus and subsequent research that put fungi on the map. And so now the state of marine fungi today is still not super impressive. We haven't really named very many species. There's about 300 named species of marine fungi, which might sound like a lot, but compare that to about 100,000 species of fungi total, you can see it's a really small percentage of these fungi.
I have to explain obligate.
Yeah. So these are fungi that are only found in the ocean. So you don't find them on land. As far as we know, they live their entire life cycle in the ocean, and we don't see them anywhere else. And so these obligately marine fungi, a lot of them have really interesting shapes and adaptations living in the ocean. So this is a species of marine fungus called Halosphaeria. This is a spore. This is one of these fungal spores, and you see it has these beautiful wings, almost like it's about to take flight. And so a lot of these marine fungi have adaptations like this that presumably help them disperse, keep them from sinking to the bottom of the ocean, and help them float around towards the top so they can find new places and access new foods. Most of the research into fungi, people are really interested in fungi for medicine. They produce a lot of chemistry, a lot of our antibiotics. Most people in this room probably owe their lives to fungi. At some point in their lives, they've taken some antibiotic. And other fungi break down very difficult substrates like lignin. So people are using fungi now to produce fuel, right? But all of that work has really been done on fungi that are found on land. So we don't know hardly anything about the potential of marine fungi to do these things. The gentleman who took this photo in the 1980s is named Jan Kollmeyer. And he is the person who really brought marine fungi back to the fore. We've got people interested in it again. And he actually did a sabbatical right here in Hawaii, and maybe some of you ran into him. And so for that reason, a lot of our marine fungi actually have Hawaiian names. So anything that he discovered from Hawaii, he put a Hawaiian name on it. So Hawaii looms large in this world of marine fungi. And a lot of his collections are deposited in New York, at the New York Botanical Garden. And maybe you've seen collections at the Bishop Museum or at University of Hawaii. They're usually pretty boring. They're pressed plants that are stuck between two pieces of paper. This collection is awesome because he was always working. And so whenever he went, he was always looking for new marine fungi. And so this is a collection that's deposited in the New York Botanical Garden. And I know this is sort of zoomed out, so you might not be able to tell what that's a picture of, but I will zoom in here. And it's a McDonald's cup. So he walked out of a McDonald's. He was in North Carolina where he lives, and he saw some sea foam, and so he scooped it up with a McDonald's cup and then went back to his lab and looked under a microscope and described 5 species just from sea foam that he scooped up in his McDonald's cup. And this is what's deposited in the New York Botanical Garden. You can find a lot of these collections online, which is pretty fun. The state of marine mycologists today isn't much better. So this is a meeting that I went to in April in Woods Hole, and this is every marine mycologist, people who study fungi in the ocean, on Earth, all standing together on the steps of the building. And this isn't just, professors. This is postdocs and graduate students and a couple of people who wandered in off the street. And this is all of us, right? So it's a really small niche group, which I think is awesome. And so we went out, we did a field trip where we got on a boat and went out to see the whole of Woods Hole, which is an interesting formation. And we decided to take 2 boats because we just said, if one boat goes down, we don't want the whole field of marine mycology to go with it. So we split up. And so we have to do sneaky things like that to keep our field afloat. Anyway, my point is not very many people are working on this. So pretty much every discovery is something brand new and pretty wild. It's a pretty wide open field, which is exciting. So why should we care? Most people are studying fungi in soils or on plants. What's so important about fungi in the ocean? Well, there's a few things that make them pretty cool. One is that fungi are amazing pathogens, so they attack living things. I have a little bit of a cold right now. It's probably caused by a virus or a bacterium, but we also find fungi that kill your tomatoes, right, that get between your toes and cause problems. They also attack phytoplankton, so our photosynthetic plankton that produces oxygen gets attacked by fungi regularly. And we might assume that if they didn't get attached by fungi, then our oceans would just be full of these phytoplankton, and they'd be a lot less fun to swim around. So this is probably pretty important for global carbon and for oxygen and for maintaining our atmosphere. We need to learn more about that. We also find that fungi dominate sediments. So one place where we find a ton of fungi is in the very, very deep ocean, 4 kilometers down below the surface. We find sediments, and in areas where there's a lot of carbon, and those are those green dots on the bottom, you see where there's more green dots, where those green dots are bigger, there's more of the red fraction on these stacked bar charts, which are fungi. So we find that fungi are really important for cycling nutrients like carbon and nitrogen in the deep ocean. Lastly, fungi play a very underappreciated role in what we call the Mycoloop. So this is a picture of a Daphnia, which is, I think they call this a sea louse. This is from freshwater, but we also find similar animals in the ocean. And that black stripe going down its middle is pollen that it's consumed. So they eat a lot of plant pollen from flowers. It turns out, though, that they have a really hard time digesting that pollen unless that pollen has been infected with a fungus. So the fungus breaks down some of those difficult to digest compounds in the pollen, and the Daphnia then is able to absorb the nutrients more readily. When we look in fish, when we look in invertebrates, we find that their intestinal tract are full of fungi. And so either those fungi are helping to digest food, or those animals are actually eating fungal spores and living off them. So fungi are a pretty important part of the food web for these really small animals that aren't eating things like fish.
May I just say something about
You may say something.
Many years ago when I went to see the salmon spawning in Canada, the professor said that the little, I don't know what you call the small ones, he said they eat the pollen from the tree, the pollen falls.
Mhm.
And when you said that was a lot of pollen,
Yeah,
I figured that's probably food.
could be, could be. So, so I don't really study marine fungi, though. This is where the bait and switch comes in. My lab is almost entirely funded to study fungi that are associated with plants. So that's where every money, all the money that pays my graduate students and my postdocs, and the machines in my lab comes from studying these foliar fungi. And this is what I was most interested in before I came here to Hawaii and got interested in the ocean. And what I study are these fungi that live in between the cells of leaves. So if you hold up a leaf that looks totally healthy, any leaf will do. It could be the Naupaka or the Vitex or anything out here, and you hold it up, you won't see anything on the surface. But if you look under a microscope or use DNA, you'll see that there's potentially hundreds of different species of fungi living inside these leaves. And so what my lab has done, we've gone all around Hawaii. We just wrapped up a big project. We studied 850 different native plants from 5 of the Hawaiian islands, and we wanted to characterize these fungi. These are some of the isolates that we can grow up in petri dishes. You can see they're all different colors and shapes and textures, different chemistry, which is important for discovering new medicines. And what we can do when we put all of this information together about how all these fungi are arrayed among all these different plants and all these different islands is we can create a model that will tell us what are the different factors or variables that shape these different fungal communities, these groups of fungi that are living in leaves or on leaves. And so you can see from this study, what we found was that things like evapotranspiration, how fast water is being evaporated into the air via leaves, is very important for determining which fungi you found in which places. How high up you are or how close you are to sea level is another important factor. And host plant phylogeny, so different plant species will harbor different fungi. And presumably those interactions are co-evolved. So you find a plant that evolves to a certain shape and to grow in a certain place. Fungi that live in its leaves have evolved along with it. And so they track each other's evolution. So we do this type of thing for fungal communities on land. And I was really interested to see whether or not we could do similar types of research on fungi in the ocean, which nobody has really tried to do before. And this all came to me, like most good ideas, when I had a cocktail in my hand at a wedding. And it was a friend of mine who studies coral reefs. He's here in Hawaii and he works at NOAA. And so he had all these people at his wedding who also study corals. And I was looking around talking to coral people, and this one guy looked at me and said, "Wow, you study fungi and leaves, huh? That's pretty cool. Maybe you've seen this paper that just came out where this gentleman, Ventus, found these fungi growing inside corals." I was like, "No, I'd never heard of that." And so this is an image from this paper, and you can see those mycelia, those ropes of fungi that are actually boring into the coral skeleton, making holes and living there. And so this came out in 2000. This was a microscopist who did this work. And this was pretty much all that he concluded from his paper, was that, wow, there's fungi in corals, but he didn't know what species of fungi those were, what they were doing with the corals. It was sort of a first pass at this system. And so I wanted to follow up on it and see who are these fungi and what are they doing? Why are they there? So I collaborated with my friend whose wedding I was at, that's Tom, and another graduate student who was here at UH at the time, Dan Barshis, and they were studying corals in American Samoa. And what they were interested in was how corals were going to survive as ocean temperatures started to rise. And so for many of you, this will be probably a familiar story. It's made a lot of news recently that our coral reefs are in trouble. And as ocean temperatures rise, there's a breakdown in the symbiosis between the coral animal and the alga that it harbors inside its tissues. So corals are we call them heterotrophs, or they're omnivores. So they get nutrients from eating different particles and plankton that float by, but they also get nutrients from these algal cells that photosynthesize and produce carbon via photosynthesis. And what happens is, as ocean temperatures rise, the algae inside these animals are no longer happy, and they leave the animal in a process known as bleaching. And some corals can recover, but a lot of corals die off in the process. And so this being an El Nino year, they're predicting is probably going to be another big bleaching year for Hawaiian corals. So people are interested in studying why this happens and how we can hopefully prevent it. And so these gentlemen had developed this great study system in American Samoa on the island of Ofu, where they have different tidal pools at the front of this island of different sizes. And so at low tide, these pools are isolated. They're surrounded by rocks. High tide comes up and they fill up with ocean water. They cool off again, and then low tide comes and the water recedes and it isolates these pools. Now, the smaller pools get a lot more sunshine and they heat up much faster. So the temperature in the small pools is much higher than the temperature in the larger pools. And because of that, you find different species of algae that associate with corals, some of which are very tolerant to high temperatures, and some of which are much more susceptible. And so what I was interested to see was whether or not fungi associated with these corals was going to be different in these two pools and with these two species of algae that were found. So we went to American Samoa, we collected a number of corals from these two different pools, one hot, one cold, and then we looked to see what sort of fungi were there.
Why
Yes?
do you say fungi and not fungi?
Because that's how you pronounce it.
Yeah.
I don't know, that's how I was taught to pronounce it. I'm sure either one of those is proper, because there's not many people speaking Latin anymore, so we can't ask them what the right way is. But
I'm looking at that.
fungi, let's call the whole thing off. So the thing about these fungi is that they're microscopic, and so you can barely see them under a microscope. But if you do see them under a microscope, you won't be able to tell what species they are or what they're doing. It's very difficult. And so we have embraced the DNA revolution. Now people are sequencing genomes and microbiomes, and very early on we saw that this is going to be very beneficial for describing diversity of these incredibly small organisms that we find in our oceans. So that's what we're doing here. We're taking little chunks of coral, little chunks, I'm talking like the size of a pinky nail. We stick it in tubes, grind it up and extract DNA. And then we use genome sequencers to sequence barcode regions of those fungi. And these machines are incredible. And so just in the time that I've been studying science, about 10 years or so, we've gone from studying, generating 100 sequences overnight to generating about 25 million sequences in a couple of days. And it's really cheap now too, so anybody can do it. It's nothing to do it. So we can grind up all these different corals from a bunch of different locations, throw them at a genome sequencer, and what we get back are these different barcode sequences that tell us what species are there, and to some degree, how many of them, what are their relative abundances in those samples. And based on that information, we can have an idea of what a community of fungi looks like. And this is a microbiome. So all of the microbes, the fungi that are contained inside these corals. And so most of the research that I'm going to show you today uses this basic protocol with DNA sequencers. And so what we found was that ecologically, there was really no difference. Hot pond, cold pond, thermal tolerant alga, thermal susceptible alga, they all had the same fungi on them, more or less. There was no story there at all. But I was interested to see what species of fungi we were going to find. And so what you're looking at here is a phylogeny, or a tree of different fungi that we found. That inner circle is just color-coded based on what class those fungi are assigned to. And then the outer circle is sort of interesting. That's what I want you to focus on here. The things that are coded in black on this outer circle here are species that we found in this study. Things that are coded in gray or in yellow on that outside circle are the closest match that we could find existing in a reference database. So there's a big database on the internet that everything that's ever been sequenced before, every mushroom, every fungus that's ever been described is added to that database. And so we searched that database for the closest relatives. So you can see there's a few narrow black bands that are sandwiched between things that people have described before, right? So this is things that are pretty commonplace and things that we see, you know, in our coffee cup or growing on our food. But where you see these really thick black bars are things that are more closely related to each other than to anything that had ever been sequenced before. So my point is there's a lot of new species, new genera, new families, deep evolutionary history that no one had ever sequenced before because no one had been doing this work in the oceans. So most of our groups were found in these ascomycetes and basidiomycetes, and these are the groups of fungi that form mushrooms. And so this, where I've highlighted here, is one of these particularly diverse, novel groups of fungi. You see there's a lot of black on that outer ring. And this corresponds to the Ustelaginomycotina. So has anybody ever had corn smut, huitlacoche? It's a Mexican delicacy. So if you leave your corn out too long, it'll develop these big black slimy pustules called smut. And so this is the group that produces smut, which was sort of unusual to find in corals. Yes?
So smut is like a fungus?
Yep, smut's a type of fungus.
Does it kill the plant, or?
It doesn't kill the plant. It grows on the corn seeds, and so it prevents it from reproducing.
Oh.
But it's usually not so devastating that it'll kill the entire plant. But it's a good question.
What is this? Mycetes and
Those are the names of different groups of fungi that we found. Those are the Latin names. So when I treat out those species, I found that they belong to this group of fungi called the Malassezia. Has anybody ever heard of the Malassezia before? Well, whether or not you've heard of it, it has heard of you, because it turns out the Malassezia is a fungus that is all over our bodies, right? So it's on your head, it's on your arms, it's on the palms of your hands, and it's really well known for causing dandruff. So this was one of the very first fungi that had its genome sequenced, and it was sequenced by Procter & Gamble because they wanted to make a better Head and Shoulders shampoo. And it turned out that the fungicides that they were adding to their shampoo weren't very effective, and so they wanted to sequence this fungus that's growing on our heads to see if they could figure out a better way to kill it. And it's a really weird fungus. It turns out it's got a tiny little genome, and it doesn't produce its own lipids. It can't produce its own fats. So it has all of these enzymes that it spits out of its body to eat your enzymes and to eat your lipids and to eat your fats. So we find it growing on our greasy bits. And so since I made this discovery, they've described a number of different Malassezia species that are associated with different animals, and they're always associated with animals. So there's one that they find on goats. There's a couple that they find on parrots. They just discovered one that grows on bats. And I found a whole bunch that grow on corals, which was so weird, and nobody expected that. And so so people started sort of scratching their heads, so to speak, because if it's something that's growing all over your body, and we're doing these very sensitive DNA-based analyses, people assumed that what we were finding was totally made up, right? And so it was sloppy lab work. Somebody scratched their head, the dandruff fell into a tube, we sequenced the DNA, and instead of actually finding something that was actually in the ocean, we were just sequencing stuff that was growing on our body, right? And so every time we get something like this, and so this is a real, you guys all, we have a president who's big on Twitter, so you guys all know what Twitter is now. This is an actual Twitter exchange from one study that came out where they found Malassezia in sponges. And somebody was like, yeah, right, no way. And it's this endless debate trying to convince people that this is for real. And so I got sick of trying to convince people that it was for real. So I wrote a paper, and I went out and I looked all over the world. I went back to this database where everything that's been sequenced is deposited on the internet. And I pulled out every Malassezia that had been sequenced in the ocean. And I made a big tree. And so what you see is that Malassezia is everywhere. We find this fungus on us, yes, but we also find it in corals. We find a lot of it in corals. We find a lot of it in sponges. We find it in these sediments. We find it in hydrothermal vents, you know, these very hot spots in the bottom of the ocean that are spewing up gases. We find Malassezia there. So this is a fungus that gets around. This is probably one of the most widespread fungi on the planet. And we know very little about it. And so this fascinated me, and this sent me down this rabbit hole ever since, trying to learn more about Malassezia in our oceans. So I'm going to refer back to this slide that I showed you before. This is the work that I did on plants. And so it's got me thinking about, well, if we find these fungi that are very widespread, that are growing on us, that are also growing on corals, I wonder whether or not these old models apply. And one thing that we find in fungi associated with plants is that the species of the host is very important. So if you study fungi that are growing on an ohia tree and compare that to fungi that's growing on a koa tree, you'll find very different fungi. And I wanted to know whether or not something similar would be found on the plants of the ocean, on algae. Bless you. So I went and did a collaboration with Dr. Heather Spalding, who was a PhD and a postdoc here at UH, and she's now a professor at the University of Charlotte. And she studies algae, and especially these mesophotic algae that you heard about last week. And what I wanted to know was that if you go down and you sample algae of different species, will you find different species of fungi associated with them? And she thought that was so cool, because she's like, you really think you're going to find fungi all the way down in these mesophotic reefs? And I said, yes, I do. She said, well, let's check it out and see. So these mesophotic reefs, as you learned about last week, are very, very unstudied. Very few people have gone and done work down there. They're at the extreme light level. They're at the darkest part of the ocean, where there's just enough life, or just enough light to sustain photosynthetic life. So you can find algae growing there, you can find corals that are photosynthetic there, but if you go just a few meters down, there's not enough light to sustain life. So life drops off. So it's similar to plants. You can find plants there, and I put plants in quotes, but the environment is completely different from any type of environment where you'd find plants on land. And so that made it very, very interesting to me to try to study what fungal life was like on these plants of the ocean. The other neat thing about these reefs is that when we think about reefs, we think about our lovely Hanauma Bay out here that's full of these gorgeous corals, but most of the reefs down in this dark twilight zone are actually dominated by algae. So you will find corals there, but most of the biomass, most of the living material is actually algae. So it's a good place to go look for algal associated fungi. The reason that it's so hard to do work down there, and I know Allison showed this slide last week, was because it's right in this reverse Goldilocks zone, right? The Goldilocks zone is the sweet spot where it's not too hot, not too cold. This is exactly the opposite. This is a place where it's impossible to go do research. If you're doing work in shallow water, you can use scuba gear, right? And maybe some of you guys have scuba dive. And you can do that up to about 30 meters comfortably, maybe down to 60 meters if you have some special training. Then, if you want to study really deep ocean, you can use a submarine. You can use a remote submersible, and that will take you down, about 150 meters and below. And they have remote submersibles that'll go all the way to the seafloor in these abyssal trenches. But that zone between 30 meters and 150 meters is really difficult. It's too shallow for the submarines for the most part, but it's too deep for scuba divers for the most part. So for that reason, this zone has been pretty much overlooked with a few notable exceptions. And we're finally figuring out how to go down there and research. So being able to get algae from this zone was a real triumph. And so we were very excited to see what we would find. The way they do that is actually a combination of using these submarines that have been very, very well trained, people that know exactly what they're doing, and novel technology that allows people to scuba dive. And they go down together. And so the submarine will grab some samples, and the scuba divers that are on rebreathers will grab some other samples. They'll all come back up and share. So this was not an adventure on which I took part. I just got the samples, analyzed them. So we analyzed samples from 4 different locations in the Au'au Channel, which is between West Maui and Lanai. And that whole red zone that you see surrounding Maui Nui is all one of the largest mesophotic reefs on the planet. So it's very extensive, and it's all at about that depth where there's still a little bit of light, but it's really hard to get at. So this is a giant mesophotic reef.
I
Yes.
think it's wonderful that you have all of the critical marks on them.
Oh, thank you.
Wonderful.
That was one of my students made that slide. I'm usually too lazy, but I appreciate it. I'm glad you noticed that. So with Heather and her colleagues, we sampled 4 different species of algae. And you can see, I don't know if anybody here is an algae expert. I'm certainly not, but these are about as different as algae get, right? You find things like Halimeda, which created a lot of the sand that you see here at Hanauma Bay. It has calcium in it. You find red algae, green algae. We found 4 very different things. And what we assumed was that if we have different types of algae, we're likely to find different fungi growing on them, was how our thinking went. And so what we found is that there was a difference between species of algae, but that was the only thing that was driving differences in fungi. So it didn't matter how deep we were, where we collected those algae, or which of those 4 reefs we were diving on. It was really just the species of algae that mattered. So all of those dots that you can maybe barely make out, they're color-coded by species. And those circles that are drawn around them are sort of the statistical equivalent of a community composition. And the only thing that I want you to see is that you find a green circle and a purple circle, and then a much smaller red circle. And so that red circle is this one species, Microdictyon, whose fungi tended to be less diverse. So there's fewer species growing on this species of algae. And wherever we collected it, it was very similar, the species of fungi that we found. So whether you were close to Lanai or close to Maui, if you pick up Microdictyon, you tend to find the same 15 species of fungi always. So again, this pattern of coevolution or of close symbiosis where these things are living together. And so that was pretty neat to find. Now, you remember that what was motivating me to study this was because I study plants in this habitat that we're pretty used to studying plants on land. And so I wanted to study the most different types of plants that I could by going to these mesophotic reefs. So I compared all of the fungi that we found growing on algae in these mesophotic reefs with fungi that I'd found growing on plants up in Mount Ka'ala. So I was involved in a study of fungi growing on leaves because this is what our endangered tree snails are eating. They crawl around on leaf surfaces and scoop up fungi and eat them. And so I'd been hired by some conservationists to study the fungal communities on these leaves to see what sort of food was available for snails. So I had this interesting data set from Mount Ka'ala, the highest point on Oahu, and I wanted to compare it to these mesophotic algae fungi. And what I found really blew me away. 27% of the species that I found growing in mesophotic algae, 150 meters, 80 meters down, were the exact same species that I found growing on plants in Mount Ka'ala, 4,000 feet above sea level on Oahu. And so what you're looking at here, the things that are coded in green on the right-hand side of that plot, these are all of my samples from Ka'ala. The things that are coded in blue are all of the samples that I took from these mesophotic reefs. And then each one of these columns is one species of fungus. And if it's light, it means we found it. If it's really bright white, it means we found a lot of it. And if it's dark, it means that it wasn't present. And so you see that there are certain species that have indicated that are very clearly mostly marine. So we find them in almost all of those marine samples that we took, and we don't find them very often in these terrestrial samples. But there were a lot of species of fungi that we found in both Mount Ka'ala plants and mesophotic algae. And I thought that was pretty crazy because nothing else on this planet can live in these two different environments, right? We don't find fish that are swimming in mesophotic reefs that are also found on Mount Ka'ala. They can't make that transition. We don't find plants that are growing on Mount Ka'ala that are also growing down in these mesophotic reefs. So there's something very special and remarkable about fungi that they're able to live in such diverse places and are able to make that transition so readily. And this is what I think is really special about studying marine fungi, is not the fact that we find new things, but the fact that we find very old and familiar things that are somehow able to tolerate these such different environments. And so this really got me thinking now about what I want to study in the future. Is precisely this, is not new things in the ocean, but old things in the ocean and how they're connected to our life on land, right? So we all know everything flows into the sea. All of our water flows into the sea. A lot of our nutrients flow into the sea. Turns out that a lot of our microbes, presumably, are also flowing into the sea. And maybe those microbes are also flowing back up onto land. So to study this, we've set up a very large-scale study up in Waimea Valley on North Shore. And this was the project that was funded recently by Keck Foundation. And with a large consortium of researchers that are interested in studying things like plants and marine biology and streams, we've set up a series of plots that go from the very top of the ridge, the Ko'olaus, all the way down to the reef fronting Waimea Bay. And we have these series of plots. And in each of these plots, we're going to sample a terrestrial site, a stream site, and a marine site. And in each of those sites, what we're doing is sampling any organism or surface that plausibly contains a fungus or a bacterium. So in their terrestrial sites, we're studying things like birds. We're catching birds and studying their feces. We're catching rats. We're studying plants. We're studying soil. We have air samplers where we're studying spores that are flying around in the air. And we're doing similar types of studies also in stream environments where we're studying fish and algae, and in marine environments as well. And what we're hoping to divine from this large study is what are the sources and what are the sinks of microbes. So if you have an area that you want to replant with native Hawaiian plants, you're going to need some of those symbiotic microbes that are important for providing nutrients, right, and protection from herbivores. Where are those microbes going to come from? Where in the environment do we find these things? And so this type of study will give us insight into that, as well as how common are these species like Malassezia that you find in so many different environments that are just readily hopping back and forth from land to the ocean. The other thing that I'm really interested in figuring out is to what extent are fungi like whales. So whales are things that evolved from mammals that were on land and then went back into the ocean and turned into marine species. It seems like maybe a lot of these fungi evolved on land and then transitioned back into the ocean. But to really get at that question, we need more sampling like this of things that are closely related on land and in sea. So I want to close my talk, and thank you for being such a patient audience, just with this idea that, here at Hanauma Bay, you probably get a lot of marine biologists, And in my department, in the botany department, we have a lot of terrestrial biologists, people who study things on land. But that dichotomy is a false one. You can't really understand ocean processes without studying land, and you can't study land processes without studying the ocean. And I think by studying these organisms like fungi that jump back and forth, we can really get at this connectivity that is so important here in Hawaii and elsewhere. Thank you so much.
Great. Does anyone have any questions?
Go ahead.
Is there any place that fungi can't live?
We have found fungi everywhere we have looked for them. They're incredible. There's fungi that will grow on your maple syrup. They find fungi in Antarctica, and they actually found a fungus that could grow on the Mars rover. So they're pretty remarkable. And what I think is so, what I think physiologically makes that possible is that their cell walls are made out of chitin, the same material that our hair and that our fingernails are made out of. And chitin's very tough. And if you compare that to our bodies, to our cells, or to plant cells that are made out of cellulose, that's much flimsier. So I think they're able to go so many places because they're really tough like that. They just have a tough coating that other organisms don't. So yeah, I think if we want to start thinking about terraforming Mars, we should start with the fungi, right? Did you have a question?
Terrestrially,
Yeah.
like we understand how mushrooms reproduce, but the sediments, what does the same fungus reproduce in marine sediments?
So how does it reproduce?
How does it reproduce?
So the main life form of fungus that we find in sediments are these yeast cells. So they're very similar physiologically to bacteria in that they don't necessarily need to have sex. They can just divide themselves and reproduce that way. So they reproduce asexually. That being said, even some of these yeasts have the genetic signature of reproduction. So at some point in their evolutionary history, they've undergone meiosis and reproduced sexually. So we know almost nothing about fungal reproduction in the ocean, as you allude to. A lot of the things that we find are sort of common, closely related to common mushroom species. But if you go snorkeling, you'll never see a multicellular fungus, right? Everything that we detect tends to be these yeast cells. So doesn't mean they can't do it. It just means that we've never seen it happening, right? And so we can collect these things that are invisible, and then if we put them on solid agar in the lab, they'll grow filamentous fungi. So there's something about being in an aqueous environment that potentially inhibits that filamentous growth phase, and probably sexuality as well. But that's something that we know very, very little about.
Because typically it goes through duality phases, right?
Yeah.
It can't be.
Right.
Somehow it doesn't.
So that's an excellent point. At the very beginning of the talk, remember I talked about Jan Kollmeyer, who is this marine mycologist, sort of the modern pioneer of marine mycology. His technique for collecting a lot of the species that he described, because this was before DNA technology was really popular, he was describing everything just based on how things looked under a microscope. He would go out and collect things like kelp or like driftwood, and he would incubate it in his lab. And some of these things, I've been reading his book recently, he'd incubate these things for years to try to coax them to form these multicellular reproductive structures that are diagnostic for naming species and describing species. But most fungi in the ocean don't have the benefit of like scientists scooping it out of the ocean and incubating it for years. So what's happening out there is really anybody's guess at this point. Yeah.
I have a question. I'm trying to visualize the ocean.
Okay.
And remember that wind
Yeah.
fungus you had and whatever. Let us say that it gives off spores.
Mhm.
I'm trying to visualize, does the spores then go into the ocean water? How easy is it for the spores to get into the air once it's in the water?
That's an excellent question. So one collaboration that I've been involved in recently is Mauna Loa. So there's an observatory up at the very top of Mauna Loa where they've been sampling carbon dioxide since the 1950s. Turns out all of those samples, when they sample carbon dioxide, passes through a little filter that they change. They want to get out all the particulate matter so they get a clean carbon dioxide reading. And they've been saving those filters in a drawer for decades. And so we thought, wow, that's sort of interesting. We'd like to take a look at those. And so we've been assaying using these DNA techniques, these filters over this time series. And so up there in the atmosphere, and this is high up, and this is 13,000 feet above sea level, right? We find things that are presumably marine species, things that we find more typically in the ocean than anywhere else on land. So there is this potential that seawater and things in it are aerosolized and then get up into the stratosphere or troposphere or the jet stream, wherever, and then they're just circulating around the planet.
So it's all over.
Yeah, whether or not those are still alive is another question. So we can, just because we can detect its DNA doesn't mean that it can still reproduce, but potentially it can.